they are talking to and presenting their work to. So that is really helpful. I really appreciate it. And now, without further ado, I'm very pleased to introduce Dr. Leonie Frauenfeld, our very first speaker today. And she's a pathologist in training in her 6th year, involved with a research group of Professor Laetitia Quantitative with a specific interest for lymphoma. So thank you so much, Leonie, for being here and for presenting your work. And you're now ready to share your presentation.
Perfect. Thank you, Selina, and thank you, Anna, for organizing this webinar. And good evening, dear audience. Thank you for participating in this webinar. And I'm very glad to have the opportunity to present our data here. In the next few minutes, I'm going to talk about diffuse large B-cell lymphomas in adult patients with an aberrant co-expression of CD10, BCL6, and MOM1, which are enriched in IRF4 rearrangements. As you all know, diffuse large B-cell lymphomas is the most common non-Hodgkin lymphoma in adults, accounting for approximately 40% of the cases. The current ICC and WHO 5 classifications both recognize large B-cell lymphomas. The most common type is the DLBCL NOS, and they both recognize special subtypes. And as you can see on the table on the right side, DLBCL NOS is a heterogeneous category, and the special subtypes are as well shown in the table on the right side. The DLBCL NOS is a heterogeneous category that encompasses morphologically, phenotypically, and clinically different diseases, and morphological variants include immunoblastic, centroblastic, and anaplastic subtypes. Gene expression profiling classifies diffuse large B-cell lymphomas according to the cell of origin into 3 subgroups: germinal center B-cell type, activated B-cell type, and unclassifiable cases, as it was shown in this study from 2002 by Rosenwald et al. From this study, the Hans algorithm with immunohistochemical stains evolved as a surrogate and the most commonly used marker in routine diagnostics for the cell of origin determination in diffuse large B-cell lymphomas. In the Hans algorithm, it's really important to stress that it should be done consecutively, and cases with CD10 positivity are straight away classified as germinal center B-cell type, regardless of the other markers. In the recent years, molecular classifications have been proposed based on translocations, copy number alterations, and mutations in diffuse large B-cell lymphomas. And these are showing the results of 2 different groups with different cohorts, but they are both coming to similar and robust results, indicating that these molecular subgroups are indeed biologically significant and relevant. 2 years later, Lacey et al. Studied a different cohort that reproduced the subgroups of the former studies using only mutational data with NGS analysis, which underlines the biologic relevance of these subgroups. Furthermore, in this study, the integration of information on morphology as well as gene expression profiling with a genetic subtype was done in order to create a predictive algorithm resulting in 7 robust groups. These subgroups are as well similar to the groups in the aforementioned studies. Thus, they are of biological importance and are potentially targetable in the setting of precision medicine. To further stress the importance of genetic alterations in the classifications of lymphomas, one of the introduced new entities is the IRF4 rearranged large B-cell lymphoma, which is frequently occurring in young patients and is involving the head and neck region, including the Waldeyer's ring. Most of these cases are of germinal center B-cell type and show a favorable outcome. In a more recent study with patients under 25 years, so children and young adults, it was shown that regardless of the GCB gene expression profile, those cases have mutations in IRF4 and NF-kappa B pathway, namely CARD11, CD79B, and MYD88. And those cases frequently show an aberrant co-expression of CD10, BCL6, and MOM1. Our aim of this study was to comprehensively characterize cases of diffuse large B-cell lymphomas in adults with an aberrant co-expression of CD10, BCL6, and MOM1. we call them a DLBCL with aberrant expression, by a FISH, gene expression profiling, chromosomal number alterations, and mutational analysis. Since we were puzzled by the phenomenon that CD10 positivity co-occurs with BCL6 and MOM1 positivity. We included 55 cases of diffuse large B-cell lymphomas, all of them CD10, BCL6, and MOM1 positive, out of 3 centers, Tübingen, Barcelona, and Arizona. All of the patients were adult patients. The inclusion criteria were the triple positive phenotype, and according to Hans' algorithm, all of these cases would have been classified or are classified as GCB type. We added as well straightforward controls, 5 GCB type DLBCL and 4 non-GCB type DLBCL according to Hans' algorithm. We investigated all cases with FISH analysis, and we were using break-apart probes for BCL2, BCL6, MYC, IRF4, IGH, and for selected cases as well for the light chains IG lambda and kappa. As gene expression profiling platforms, we were using nanostring Lymph2ZX assay and or the HTG EdgeSeq DLBCL cell of origin assay. For mutational analysis, we used 2 custom panels using first of all the SureSelect with 66 genes and the IonTorrent panel with only 10 genes for verification. To predict the molecular subgroup, we used the algorithm by Wright et al. I want to quickly comment on the HTG method. Sebastian is later on going to go a bit into depth on that. HTG method is a quantitative nuclease protection assay, and the method prepares DNA library for NGS sequencing. The protection probe binds specifically to the RNA target. After that, unbound RNA and probes are digested. Target RNA is eliminated and PCR-based adapter and tag addition is done, and the probes are then quantitated, pooled, and sequenced on IonTorrent or Illumina platform. We used a special assay, as I earlier mentioned, the DLBCL cell of origin assay, which covers 64 lymphoma markers and 16 drug target markers, including BCL6, IRF4, and MME, which encodes for CD10, which was very important, of course, in our study. The HTG workflow is quite time and tissue efficient and is a robust method. The integrated RNA extraction approach prevents the loss of RNA during extraction and an easy preparation. The COO analysis is integrated within the host software, and the output will be ABC, GCB, and unclassifiable, but Sebastian is commenting on that as well. I want to quickly introduce as well the nanostring technology, which we as well used. It is a method based on highly multiplex detection of mRNA targets. The extracted RNA is hybridized with a biotin-labeled target-specific capture probe, and these probes enable stable binding to the streptavidin-coated cartridge. The reporter probe contains target-specific fluorophores, which can be digitally detected by an automated fluorescence microscope, which is seen here. Here I want to give a practical overview over both platforms. Nanostring needs an RNA extraction beforehand, whereas this is integrated in the HTG protocol. For HTG, more hands-on time is needed, and NGS facilities are necessary for the analysis. The software for evaluation is included for both platforms. Here I'm going to show the results. I'm giving overview over the clinical and morphological data. We saw a slight preponderance of female patients with a ratio of 1 to 1.4, and the median age was 70 years, ranging from 37 to 92 years. We found 32 extranodal cases, 9 with the tonsils and Waldeyer's ring involvement. Clinical information was obtained for 45 cases. 26 of the cases showed high-stage disease, and 8 patients unfortunately died of disease. For 29 patients, there was no evidence of disease and a median follow-up time of 36 months. Morphology-wise, we saw most of the cases with at centroplastic cytology, which is shown in panel B, and only 6 cases with the blastoid cytology, which is shown in panel A. FISH analysis was done and in 27% of the cases there were found no alterations, and in about 70% of the cases we found one or more alterations, including BCL2 and BCL6, each with 12 cases, IRF4 and MYC, each of 11 cases. IGH was found in 23 cases, and for the light chains it was 4 cases in total. We investigated BCL2 and MYC additionally and specifically, since these two translocations would exclude the diagnosis of IRF4 large B-cell lymphoma. With that, I'm coming to two special cases of this cohort, case 25 and 26. And I want to point out those two cases which showed a complex translocation, and both of them involved the IRF4 gene. Case 25 with the rearrangement of BCL2 and IRF4 that co-localizes with the IGH locus in the same derivative chromosome, suggesting that this is a T6-14-18 translocation as a one-hit translocation. Case 26 with IGH and BCL2 with a typical pattern of translocation and IRF4 rearrangement that is assumed to be an independent alteration. Both of these cases do not qualify for a diagnosis of IRF4 LBCL, since they have other FISH translocations. I now want to compare the two platforms we used for the cases. The gene expression profiling in total was performed in 54 out of 55 cases. 48 of the cases were investigated with Lymph2ZX nanostring assay, and 24 cases with the HTG EdgeSeq method, and 18 cases were investigated by both techniques. In about 60% of the cases, we found a GCB type, and in about 30% of the cases, we found an ABC type, and in a small subset, we found cases with unclassifiable phenotype. We saw a good correlation between the two platforms. 70% of the cases had a perfect correlation, and there were no discordant cases between Lymph2ZX and HTG EdgeSeq. But 5 cases that were classified as unclassifiable in the Lymph2ZX panel were classified as GCB-COO or ABC-COO within the HTG EdgeSeq. This is suggesting that the Lymph2ZX platform is maybe more stringent and is putting those cases rather into unclassifiable if they are not clear to the algorithm, and that the HTG EdgeSeq is rather pushing the cases into one of the two categories. A total of 392 driver mutations in 55 analyzed cases was found, with a mean of 10.6 mutations per case. And the most recurrently mutated genes were KMT2D and PIM1, followed by MYD88 and CRABP. IRF4 mutations were exclusively identified in cases with an IRF4 rearrangement, and these mutations can be observed in the C-terminal region, in the N-terminal region of the gene, which is most probably the pattern of an aberrant somatic hypermutations. In DLBCL without IRF4 translocations, IRF4 mutations can be observed, but rather in the C-terminal region. Coming back to our small table about the gene expression profiling comparison, we finally decided to classify those cases with unclassifiable and GCB or ABC profile according to the HTG results as ABC or GCB, since as well the mutational profile was demonstrating concordant results for those cases that fit quite well with the COO determination by HTG. This oncoprint summarizes the complete results and is grouping them according to the FISH results. Group number 1 is without any alterations. Group number 2 shows cases with IRF4 rearrangements. 5 cases are rearranged with IGH and 2 cases with IGL. This group also includes 2 cases with cryptic IRF4 translocations, both with an identified break in the light chains and with IRF4 mutations, and as well with a characteristic morphology. Group number 3 contains cases with alterations in BCL2, BCL6, and MYC, and the 2 already mentioned cases with the complex alterations involving IRF4. By molecular clustering according to Wright et al., 40 cases are assigned to a group. Group 1 and 2 most frequently assigned to MCD, and group 3 is most frequently aligned to EZB BN2. To summarize again, this is again the oncoprint. Group number 1 contains cases without translocations and is mostly, those cases are mostly of ABC-MCD phenotype. 70% of the group 1 are of ABC-MCD phenotype. Group 2 contains cases with IRF4 alterations and showed 50% each GCB and ABC-MCD, so it's 50-50 in this group. Group 3 contains cases with translocations in BCL2, BCL6, and MYC, and the 2 already mentioned cases with the complex translocations, and they are mostly of GCB BN2 EZB profile. We also analyzed the main pathways used by the tumors, and independently of the groups, most cases showed alterations in epigenome and chromatin modifiers. The IRF4 altered group showed significantly higher alterations in genes involved in the NF-kappa B pathway, namely CARD11, CD79B, and MYD88, and as well in B-cell differentiation. The genes altered in NF-kappa B pathway, I already mentioned in the beginning, in the introduction, that they are as well mutated in the LBCL IRF4 of the childhood. One of the questions is precisely whether LBCL in the childhood is the same disease as in adults, and therefore we compared our data with that of Rami Saldiva, earlier mentioned. No differences in recurrent copy number altered regions are found, but adult cases have a higher genomic complexity with 16.85 alterations per case in adults versus 6.25 alteration per case in pediatric cases. This phenomenon is not statistically significant. The most recurrent alteration was a copy number loss of 17p13.2, detected in 70% of the cases, including the TP53 locus in all 5 cases. However, no p53 mutation were identified. We found as well a higher mutational load in adult cases, 10.7 versus 4.7 mutations per case, with a higher frequency of KMT2D, MYD88, and BTG2 mutations, which is statistically significant. As well, we found that in adult cases, we found a lot more ABC gene expression profiling cases compared to the cases of the childhood. As a control group, as earlier mentioned, we added 9 straightforward DLBCL cases, 5 of them with GCP phenotype according to Hans algorithm and matching gene expression profiling, translocation, and mutations, and 4 cases with non-GCP phenotype according to Hans algorithm with matching gene expression profiling and mutations. Gene expression profiling was performed by HTG and nanostring and showed 100% correlation in these cases. To summarize our findings, our multi-platform approach to investigate diffuse large B-cell lymphomas with aberrant expression found 3 distinct groups. Group number 1 includes cases without chromosomal translocations and despite expressing CD10 often show gene expression profiling and or mutational profile of ABC-MCD subgroup. Group 2 includes cases with only IRF4 translocations with an aberrant somatic hypermutation pattern of the IRF4 mutations. Group 3 includes cases with 1 or more translocations in BCL2, BCL6, and MYC, and are assigned mainly to the GCB-EZB group. We thus conclude that the DLBCL with aberrant expression of adults are a heterogeneous disease which is enriched in large B-cell lymphomas with IRF4 alterations, 20% of the cases in our cohort, and frequent IRF4 mutations. We believe of cryptic IRF4 translocations, all of them with IRF4 mutations. And we identified cases with Ig lambda involvement in the translocation. Compared to children, genetically, LBCL IRF4 in adults seem to be the same disease but have a higher genomic complexity and are more often of ABC type. IRF4 rearrangement can also be detected together with a BCL2 rearrangement, but most probably this is representing a different phenomenon. And to state something very important here is that DLBCL with aberrant expression are not necessarily of GCB type in gene expression profiling, and it might have been suggested by the Hans algorithm. To conclude, among the DLBCL cases with aberrant expression, 40% of the cases are indeed misclassified as GCB according to Hans' algorithm. And mutation analysis and gene expression profiling give complementary information and demonstrate further the heterogeneity of diffuse large B-cell lymphomas with aberrant expression. The HTG, EdgeSeq, and nanostring technologies showed a good correlation and direct comparison. 5 out of 18 cases were unclassifiable with nanostring. Those cases were either ABC or GCB type in HTG EdgeSeq platform. But I want to stress here that this study was not designed to compare the two gene expression profiling methods, but we saw a good correlation and direct comparison. Again, I want to stress nanostring seems to be more stringent in the classification of cases, and HTG rather tries to push the cases into one of the categories ABC or GCB. Finally, I want to state that the DLBCL with aberrant expression of CD10, BCL6, and MOM1 should not be classified with the Hans algorithm, since this is a very heterogeneous group of cases. With that, I want to thank you for your attention. And of course, I want to thank all the collaborators here in Tübingen, in Barcelona, and Arizona for the nice work we've done together. And of course, to our principal investigators. And now I'm giving back to Selina, and I'm happy to take your questions after Sebastian's talk. Thank you.
Thank you so much, Leonie, for this wonderful presentation. And I'm sure the audience has some interesting questions for you. They need to wait for the end. After Sebastian's talk, we will have all the questions answered together. So please use the chat function to type in your question. You can already type it in there now so that we have it ready at the end for the open discussion and the Q&A. So it's my honor to introduce Sebastian Streich. He's a medical student in his last year, and he's also working in the field of hematopathology in the doctoral thesis. He is also involved in the research group of Professor Laetitia Quintanilla-Fend, and so I'm very pleased that he's now complementing the study that was done here also on the aberrant expression studies on large B-cell lymphoma. So Sebastian, you can now share your presentation.
Thank you, Selina. You should already see my slides. Do you see them? Yes, thank you. Okay, perfect. So, dear colleagues, dear ladies and gentlemen, it's a pleasure to present our latest research in today's HTG molecular webinar. Fortunately, we were able to publish this research earlier this year, so most information I'm going to show is also available in our publication. Let me just start with a brief introduction. Leonie already gave an overview about the classification of the B-cell lymphomas, so I am just going to highlight some important facts of my topic. The latest classification systems of hematolymphoid tumors by the International Consensus Classification and the World Health Organization in the 5th edition are congruent in the most parts. However, there are some small differences in some distinct features. And first up, I'm going to show the ICC classification. Within the group of the mature B-cell lymphomas, there are several subgroups, including the diffuse large B-cell lymphomas, and not otherwise specified, which are the most common B-cell non-Hodgkin lymphomas in adult patients, as well as large B-cell lymphomas with IRF4 rearrangement or LBCL IRF4. Furthermore, there are the follicular lymphomas, as well as other entities. DLBCLs are subclassified according to their cell of origin, or COO, using gene expression profiling or the surrogate marker immunohistochemistry. And the subtypes are the germinal center B cell, or GCB, and the activated B cell, or ABC subtype. Follicular lymphomas are graded histologically into grade 1 to 3B. It is known that rearrangements of the IRF4 gene occur most commonly in DLBCLs, and here first and foremost in GCB type DLBCLs, as well as in follicular lymphomas grade 3B. These rearrangements led to a reclassification of those cases as cases of the new entity of large B-cell lymphomas with IRF4 rearrangement. The WHO classification, on the other hand, defines in the group of the mature B-cell lymphomas several subgroups, including the large B-cell lymphomas as well as the follicular lymphomas, among other entities. The group of the large B-cell lymphomas consists of the DLBCLs as well as the large B-cell lymphoma with IRF4 rearrangement and other entities. The WHO classification also uses the COO to further distinguish the DLBCLs. In the group of the follicular lymphomas is again the entity of the follicular lymphomas, which has several subtypes, including the large B-cell lymphoma, the follicular large B-cell lymphoma, or FLBL, which was named as follicular lymphoma grade 3B in the 4th edition of the WHO classification. Also here, IRF4 rearrangements are known to be most commonly in GCB-type DLBCLs as well as FLBLs. So both classification systems are congruent in the most parts, including the definition of the large B-cell lymphoma with IRF4 rearrangement as a distinct entity. And next up, I'm going to focus on this new entity. The gene IRF4, or interferon regulatory factor 4, encodes for the protein MAM1, which consists of 451 amino acids. And IRF4 MAM1 is known for numerous interactions with other transcription factors, including those of the NF-κB pathway. And it could be shown that IRF4 deficient mice do not produce any B-cell lymphocytes, which shows the important role of IRF4 MAM1 in the B-cell development. IRF4 MAM1 is expressed in the majority of B-cell neoplasms, including DLBCLs. In many ABC-type DLBCLs, the NF-κB pathway is constitutionally activated, which again activates IRF4 MAM1. And in a subgroup of the GCB-type DLBCLs, a translocation of IRF4 into the gene locus of the heavy immunoglobulin chain IgH activates the expression of IRF4, which also defines the new subtype of LBCL IRF4. This new subtype shows some distinct features in comparison to regular DLBCLs and follicular lymphomas grade 3B. First of all, the characteristic IRF4 mutation, which defines this entity, can be found in 2 to 21% of all large B-cell lymphomas. Those cases with the translocation of IRF4 are most likely to also carry those cases with the translocation of IRF4 are most likely to also carry IRF4 mutations due to the translocation into the IgH promoter and the consecutive aberrant somatic hypermutation. Most of the LBCL IRF4 cases show a GCB phenotype, which is also explainable by the pathogenesis of these cases in the germinal center. And the translocation of IRF4 into the IgH promoter region leads to a consecutive expression of MAM1 in the majority of the cases, which is an uncommon feature for GCB-type lymphomas. In many of the LBCL IRF4 cases, mutations in genes of the NF-κB pathway can be detected, for example, MYD88, CD79B, and CART11, as Leonie already mentioned. Several studies have shown an accumulation of LBCL IRF4 cases in children and young adults, which shared an excellent prognosis. Finally, it has been shown that LBCL IRF4 most commonly occurs in Waldeyer's ring, as well as in the GI tract. However, the frequency and the features of LBCL IRF4 are not well known for adult patients. Therefore, we designed this study to comprehensively characterize cases of DLBCLs and follicular large B-cell lymphomas or follicular lymphomas grade 3B of the Waldeyer's ring in adult patients, using a complementary approach to determine the prevalence and characteristics of LBCL IRF4 in adult patients and to compare these lymphomas in Waldeyer's ring with those of other locations. So now I'm going to give you an overview of the methods we used, including the gene expression profiling using the HTG EdgeSeq system. Let me just briefly talk about our case selection criteria. We screened the archive of the Institute of Pathology at the University of Tübingen for cases of de novo DLBCLs not otherwise specified and follicular large B-cell lymphomas. These cases needed to occur in adult patients and had to be localized in Waldeyer's ring. And for the subsequent analysis, sufficient FFPE material was needed. These criteria led to 30 cases, which had been diagnosed between 2002 and 2020. As a first step after the inclusion of the cases, we performed several immunohistochemical analyses, including CD10, BCL6, and MAM1, forming the Hans algorithm, which is a surrogate marker for the cell of origin. And furthermore, we examined the immunohistochemical status of BCL2, MYC, and Ki67. Next up, all cases underwent comprehensive fluorescence in situ hybridization or FISH analysis using IRF4, BCL2, and IgH gene probes provided by Cytovision and Abbott Molecular. Moreover, all cases were analyzed using a next-generation sequencing workflow on the Ion Torrent S5 platform. Therefore, a custom AmpliSec DLBCL panel was used, which included IRF4, which is known to be frequently mutated in LBCL IRF4 cases. Furthermore, EZH2 and BCL2 were analyzed, which are known to be mutated especially in GCB-type lymphomas, as well as MYD88 and CD79B, which are first and foremost mutated in ABC-type lymphomas. Additionally, 5 more genes were investigated for mutations, and 5 of those genes already mentioned were additionally examined using single amplicons due to bad coverage in the NGS panel analysis. Finally, I'm going to talk about the gene expression profiling using the HTG EdgeSeq system. We use the HTG EdgeSeq DLBCL cell of origin assay for Ion Torrent platforms, which contains 92 transcripts of frequently affected genes and their transcripts in DLBCLs, including the genes encoding for the proteins of the Hans algorithm, as well as BCL2 and MYC. And the EdgeSeq workflow was already mentioned by Leonie, but I'm going to focus on some specific parts. The workflow started with the sample preparation, where we measure the tumor area in the FFPE material to calculate the correct amount of lysis buffer and proteinase. And then the sections of FFPE material were created and transferred to reaction tubes. Afterwards, the calculated amounts of lysis buffer and proteinase were added, and the so prepared tubes were incubated at 50 degrees Celsius. Next up, the samples were transferred to a 96 well plate for the processing in the HTG EdgeSeq processor. Using the software provided by HTG, we created a run protocol for the EdgeSeq system and loaded the system with the samples, the pipettes, and the reagent trays. And then the automated QNPA assay workflow started, and the HTG EdgeSeq system performed automatically the library preparation overnight. The next morning, the samples had underwent a QNPA, and all of the sample DNA and RNA was digested. And instead, the wells now contained the gene probes that correlated with the amount of RNA in the initial sample, as Leonie showed in the scheme in her presentation. These gene probes were then further processed using a PCR workflow for adding the NGS barcodes, and then cleaned up using an MPure XP magnetic beads cleanup system. Thereafter, the so prepared libraries were quantified using a qPCR workflow in the light cycle system by Roche. And finally, a run plan for the Ion Torrent system was created, and the libraries were pooled according to the calculation using HTG software, which I'm going to show in a minute. After completion of the sequencing, the data was downloaded and further processed, and I'm going to show these steps in a minute. Here you can see 2 screenshots of the HTG EdgeSeq library calculator, which uses the qPCR concentration as input values and calculates the amount of each sample to be added to the final library pool, so that all samples share equal concentrations in the final library pool. The Ion Gene Studio S5 Prime generates, beside other output files, FASTQ files, which contain the sequencing data in a specific format. These FASTQ files were downloaded and then transferred to the EdgeSeq parser software, which uses an HTG-developed algorithm to determine the cell of origin, and then outputted the COO classification as GCB type, ABC type, or as unclassifiable. Furthermore, the EdgeSeq parser converted the information of the FASTQ files into raw expression values, and these raw expression values were then transferred to RStudio for a quantile normalization and then used for statistical analysis. Now I'm showing you the most important results of our work, starting with the clinical pathological features. The 30 cases we analyzed shared a median age of a diagnosis of 69.5 years with a range of 37 to 87 years. And most of the cases were DLBCL type, but 2 cases were diagnosed as follicular lymphoma grade 3B. In only 23 of the 30 cases, clinical information was available, and most of the cases showed a low clinical stage, and most of the patients received R-CHOP chemotherapy, leading to a complete remission in the majority of the cases. Those of you who already read our publication will know this figure. It summarizes the results of the COO classification, the immunohistochemistry, the FISH analysis, and the NGS, and I'm going to explain this figure and the corresponding results step by step. Up first are the results of the COO classification using gene expression profiling and immunohistochemistry shown. Using gene expression profiling, 14 cases each were classified as GCB and ABC type, and 2 cases remained unclassifiable. Using immunohistochemistry, 18 cases were classified as GCB type, and the remaining 12 cases as non-GCB type. The overall concordance of the COO classification using gene expression profiling and IHC was 83%, and the COO classification led to a subgrouping of the cases into 3 groups. The first group consisted of all 14 cases classified as GCB by gene expression profiling. All of those cases were also classified as GCB type using immunohistochemistry, and most of them were positive for CD10 and BCL6 while lacking MAM1 expression. In 2 of these cases, IRF4 rearrangements were detected, which were the only IRF4 rearrangements in the whole case collective, as well as BCL2 rearrangements in 6 cases. And all cases with those rearrangements also carried mutations of the corresponding gene. Furthermore, frequent mutations were found in EZH2, CART11, and PIM1. The second group included all 14 cases classified as ABC by gene expression profiling. All of 3 of these cases were also classified as ABC or non-GCB using immunohistochemistry, and most of them were positive for MAM1 while showing a negativity for CD10 in most cases. Using FISH, no rearrangements of IRF4 or BCL2 were detected in those cases, and frequent mutations in this group were found in CD79B, MYD88, and PIM1. The third group encompassed the 2 by gene expression profiling unclassifiable cases, and those cases shared in the immunohistochemistry a ABC and GCB subtype in equal proportions. However, characteristic rearrangements or mutations were not found in those cases. However, 1 case carried a non-canonical MYD88 mutation, shown here in yellow. And now I would like to highlight some special cases. First up are the cases of the large B-cell lymphomas with IRF4 rearrangement. And those cases showed a GCB phenotype by gene expression profiling and immunohistochemistry, and also carried IRF4 mutations, which showed an aberrant somatic hypermutation pattern. The patients of those 2 cases were 37 and 78 years old. And next up are the cases with an aberrant immunohistochemical expression of CD10, BCL6, and MAM1. Leonie's presentation was all about cases with this pattern, so I'm just going to highlight the most important features. All of those cases were classified as GCB using the immunohistochemistry, hence the CD10 positivity. However, only 3 of those cases were classified as GCB by immunohistochemistry. By gene expression profiling, excuse me. Both cases of large B-cell lymphomas with IRF4 rearrangement were to be allocated to this group. And in one of those cases with a mismatch in the COO classification, a canonical MYD88 mutation could be detected, shown in green on the right-hand side. After showing you the highlights of all methods, I'm going to share some of the specific results of the gene expression profiling using the HTG EdgeSeq platform. First of all, this graph shows all transcripts with significant differences in the gene expression levels between the GCB and the ABC subtype. On the left-hand side are the genes with a high expression level in GCB type lymphomas, and on the right-hand side are the genes with a high expression level in the ABC type lymphomas. Interestingly, BCL6, MME, and IRF4 are among those 29 transcripts, which are the transcripts for the proteins used in the HANS algorithm. And these box plots show the differences in the gene expression levels sorted by the immunohistochemical status of the corresponding protein for MME and CD10, BCL6, and IRF4 and MAM1. And in all 3 transcript protein pairs, we were able to show significant differences in the gene expression levels divided by the immunohistochemical status. Finally, I would like to share a critical look on our results. And our results confirm the classification of large B-cell lymphomas with IRF4 rearrangement as a new entity among DLBCLs and follicular large B-cell lymphomas. The prevalence in our case collective in the Waldeyer's ring is 7%, and both cases show a GCB phenotype as well as IRF4 translocation and mutations while lacking other characteristic mutations. Hence, we were able to confirm the findings on prevalence as well as clinical and genetical features from other studies in children and adults in and outside the Waldeyer's ring. 28 of 30 cases were assigned to a cell of origin using gene expression profiling while showing equal proportions of GCBs and ABCs. In the literature, our GCB rates reported from 40 to 62%, so this matches our data quite well. Cases which are unclassifiable by gene expression profiling are a challenge. So in this case, a comprehensive approach using immunohistochemistry, FISH, and next-generation sequencing could provide a benefit in diagnostic or research applications. Using immunohistochemistry, the majority of the cases was assigned to a GCB subtype. The overall concordance of the COO classification using immunohistochemistry and gene expression profiling was good. The routinely used immunohistochemistry is therefore a robust yet simple alternative to the more complicated and time-consuming gene expression profiling. However, there are some limitations of the COO classification using the surrogate marker immunohistochemistry. First up are the cases with an aberrant expression of all three immunohistochemical markers, and those cases are assigned to the GCB subtype, hence the CD10 positivity. However, 2 of those cases were assigned to the ABC subtype using gene expression profiling. Furthermore, 1 of those cases showed a canonical MYD88 mutation supporting the ABC subtype. Hence, cases with aberrant expression of all 3 immunohistochemical markers should not be classified using immunohistochemistry. 5 cases showed a non-matching COO classification using gene expression profiling and immunohistochemistry. However, 2 of those cases were the already mentioned unclassifiable cases, and 2 more of those cases showed an aberrant expression of CD10, BCL6, and MAM1. And overall, a comprehensive approach is needed to correctly classify those cases. The gene expression profiling workflow by HTG Molecular is an easy-to-use method for NGS-trained personnel. Using almost 100 transcripts, the COO classification gets a robust molecular basis. The routine workflow using the EdgeSeq parser is a fast and simple way to get the COO classification. However, the complete workflow is more time and resource consuming than the established immunohistochemistry. And the statistical analysis using the raw expression values are a valid method for research application, but due to the high bioinformatical workload, not for routine applications. Using next-generation sequencing, some mutations could be identified, which are already known to be used in novel classification systems. However, those studies use much larger NGS panels, so the panel used in this study only shows limited significance for this application. Besides that, some of the cases showed characteristic mutations that are known to occur in defined subgroups, which reinforces the classification using immunohistochemistry and gene expression profiling. In conclusion, large B-cell lymphomas involving the Waldeyer's ring in adult patients were equally divided into ABC and GCB subtype and showed some characteristic molecular features. The overall concordance between gene expression profiling and immunohistochemistry was excellent, except for cases with aberrant expression of CD10, BCL6, and MAM1, and unclassifiable cases. LBCL IRF4 in adult patients shares clinical, morphological, and genetic features with LBCL IRF4 in pediatric populations. However, this relatively small cohort in adults suggests a lower prevalence of those cases in Waldeyer's ring in adults compared to pediatric patients, which needs to be confirmed in a larger cohort. Last but not least, I would like to thank all members of our research group for their contribution to this work, as well as the team of our technical assistants for their excellent work in the lab. I'm now pleased to answer your questions and hear about your thoughts in the, about our presented work in the discussion. Thank you for your attention.
Thank you so much, Sebastian, for this wonderful presentation. Really interesting insights that you provided there. So I invite the audience to use the chat functions to ask any questions. And in order to give you some more time to ask some questions, I'd like to start with some questions for you both. So the first question is for you, Leonie, about the 3 molecular different clusters that you identified in the DLBCLs in adults with aberrant co-expression of CD10, BCL6, and MAM1. So do you have future plans to further validate these clusters that you identified? And do you think, based on the molecular data that you discovered, that they could benefit from different treatment strategies?
Definitely, that's a very good question. Thank you, Selina. So, of course, this was a very small cohort with only 55 cases. So, of course, it would be crucial to add some more cases to this cohort, and this would be of major interest. And of course, these subgroups, which are now defined, not only the IRF4 LBCL, but as well the other subgroups by the algorithm by Wright et al., they all can lead to a precision medicine and to drug targets, and that's really the importance of what we are doing now, to specify this very heterogeneous disease of DLBCL, and to get more insight into the molecular background. And the aim of it is, of course, to identify targetable drugs. And yes, that's a good question.
Thank you. And that brings me right to my next question, because I think both of you showed the molecular heterogeneity of DLBCL, and at the same time, in particular, Sebastian, you presented also some good correlation with immunohistochemistry being still the gold standard for many of the markers that are identified. So very broad question, how to bridge that gap? Ideally, you should get as much data as possible, but still it needs to be clinical applicable. How do you see that in particular for the subgroups that you identify, how to best approach it?
Would you like to answer, Leonie, or should I answer?
You can answer.
Okay. Yes, the data I've shown suggests that the molecular subgroups are or suggest different subgrouping besides the immunohistochemistry. However, this is not currently shown in the routinely diagnostics. And you mentioned the gap between the research and the diagnostic applications. So I think what has to be done is to develop easy and or easy to use and time efficient workflows to bring those statistical analysis I've shown and so on to a routine workflow, so that anybody with not the best bioinformatical background can do this analysis, and this I think would provide a excellent benefit for the diagnostic approach.
And if I might add something to that, I think what is really crucial and what our studies show is that for cases that show somehow aberrant phenotypes, it's very important to go more into detail for those cases. So the straightforward cases, as well as I've shown in my control cases, those cases are the rather easy cases. But those cases which have an aberrant phenotype, those cases should be investigated a bit more closely.
Okay, thank you both. So I look in the chat window and we have some questions from the audience. One question is, does the HTG EdgeSeq DLBCL cell of origin assay use only gene expression to assign molecular subtypes, or does it incorporate mutational data as well? So I think I can answer that. The HTG assay only does gene expression profile. There is no mutational data included, so only the expression levels of the genes. But there's another question for you, Leonie. When comparing the cell of origin by nanostring and HTG, you mentioned the HTG method is less stringent and therefore results in fewer unclassified calls than the Lymph2CX. A column in the table of results on slide 22 shows the cell of origin final column corresponding to the HTG calls. How was the final call determined?
Nice questions. Thank you. So the final call was determined using the molecular data as well, so the mutational data. The mutational data were very much fitting to the HTG method, and that is why we decided to not put them into the unclassifiable category as proposed by the nanostring prognostication, but to put them into the HTG method-based COO. So we compared it with the mutational data and had this approach to this problem.
Okay, thank you for answering these questions. So currently we don't have any other additional questions, but in case there are questions coming up, feel free to reach out. And also if you're watching the recording, we are pleased to forward the questions to the speakers and follow up after the webinar with questions that might still come up. So with that, I would like to really thank our speakers for the excellent presentations and of course the work that you put into, and really wish you all the best on the continuation of investigating the complexity of these DLBCLs and therefore also revealing some really valuable insights that deserve the patients and help to derive better treatments. So thank you so much.
Thank you.
Thank you.